So we've looked at DNA replication, which when does DNA replication happen in our stages of our cell cycle? It's in the S phase. The synthesis, what is being synthesized is our new strand of DNA. So that's the S phase. But the G or growth phases are basically the gaps between mitosis and the synthesis of our new strand of DNA. So we're going to see for mitosis, we're going to break down the nuclear membrane. We're going to lose the nucleolus as the chromosomes start to condense and that material spreads out through the cell. So that's where we're going to recreate our nucleus at the end of mitosis. That's going to migrate to its position in the cell during our G1 phase. But the biggest thing of G1, the cell's going to grow. Because think about it. When you take a parent cell and you produce two daughter cells, they have the same genetic composition, but what's the size difference? Each daughter cell is half the size of the parent. Now, if you do that often enough without growth in between, you're going to end up with some cells after several rounds of division that are teeny, teeny, tiny. So after the cells divide, they have to grow. And you have to produce more organelles because what did the organelles do? They were split in half. So you've got to divide and make more mitochondria. You've got to make more Golgi. You've got to make more endoplasmic reticulum. So that's all happening in G1. The S phase, that's our DNA replication, 19:1. But our G2, this is where we are preparing for mitosis. G2 is immediately preceding mitosis. And so as we look at cell division and mitosis, the cloning of one cell into two, we're going to see we go through various stages as illustrated here in this animation. We're going to talk about many of these structures, but you're probably familiar with these. You may have even touched on these in high school biology, by and large, in the stages of mitosis. So we're going to go through this process in its formation. Now, when we come out of interphase, G1, S, and G2, the first phase of mitosis is called prophase. Now, we're going to have four stages of mitosis: prophase, metaphase, anaphase, and telophase. The way you remember those is PMAT. Go ahead and write that down somewhere, PMAT. PMAT, prophase, metaphase, anaphase, telophase. That's the sequence of those four major stages of mitosis. And so when we look at prophase, we're going to see that our chromosomes condense. Now, even as early as this prophase stage, our chromosomes are actually two sister chromatids that are attached at the center. The center of each chromosome being called the centromere. So after we come out of the S phase, we have our copy of our chromosomes. They're just tied together in those X-shaped structures that we recognize. So those are our sister chromatids. So our sister chromatids begin to condense. Now, don't confuse the centromere, the center part of our chromosome, the connected part of our sister chromatids, with centrosome. Our centrosome is going to be our centrioles, and these are going to be the structures that are going to organize our cytoskeletal components that are called microtubules. So our microtubule organizing center, which basically is comprised of our two centrioles, are often referred to as our centrosomes. These are going to begin to form. And these are going to begin to move toward opposite poles of the cell. Now, except for the microtubules, by and large, our cytoskeletal elements are going to begin to dissolve. These polymers are going to begin to break down into monomers as we begin this mitotic activity. Now, remember I said our stages are PMAT. A lot of folks are adding in this pro-meta. I hate that. So we're not going to do pro-meta. So when we see the nucleus disappear, the nucleolus is going to go away. Our centrosomes, these are going to be, again, our centrioles. They're going to move apart and they're going to end up at opposite poles. And as they do, these microtubules are going to be organized into this bird cage-like structure that is called the spindle apparatus. And our microtubules are called spindle fibers. Now, the reason that these microtubules and the spindle fibers are so important is some of these spindle fibers are going to attach to the centromere of our sister chromatids. They're going to be proteins that are assembled. You see the green dots in the middle of our sister chromatids? These proteins are going to assemble, and the microtubules, the spindle fibers, are going to attach, and that's referred to as the kinetochore. What does kinetics refer to? Movement. So it's going to be the connection of these microtubules, our spindle fibers, to the sister chromatids that are going to be able to move the sister chromatids where we want them to be. And eventually, the two spindle fibers attached to the centromere, it's going to be responsible for pulling the sister chromatids apart into individual single chromosomes. So do you see the movement that's taking place? At first, we're moving the sister chromatids where they need to be, and where they're going to need to be is right down the middle of the cell at the equator. And then these spindle fibers composed of microtubules are going to be responsible for pulling the chromatids apart.
So if you don't like prometaphase, what should we call this little part?
This is still part of prophase.
Okay.
We're still in prophase. So for our pro-meta, understand some people may make that designation, but I'm not going to. I still like prophase. Because metaphase, one thing is going to happen at metaphase. It's one characteristic, and if you don't see that, it's not metaphase. So I don't even like putting that in there.
So that right now is, so we're looking at
Prophase. You could call this late prophase.
Okay.
Early prophase versus late prophase are just simple prophase.
You see, I thought we were looking at metaphase and they decided to.
See, and that's it. That's why it gets confusing. Yeah, and that's why I'm not going to do it. I'm not going to confuse you. We're still in prophase.
Okay.
Because you don't arrive at metaphase until we line our sister chromatids up right along the equator, right along the midline. When you see them all lined up, like that illustration, that's metaphase. If they're not lined up, it's not metaphase. So this gives a little bit more about our kinetochores. We can see our kinetochore proteins attached at the end of the microtubules, our spindle fibers. There's our centromere of our sister chromatids. And again, as they're working in unison, they're going to move that sister chromatid back and forth. But then when they start going in opposite directions, do you see how it's just going to pull them apart? And that's what's going to happen with our kinetochore microtubules. Now, we have other spindle fibers that are simply connected from one centrosome toward the midline. They don't interact with the chromosomes, but they interact with the spindle fibers from the opposite centriole. And we're going to see that in the illustration. These are called polar microtubules. Those polar microtubules, as they extend, as we get toward the end of cell division, they're going to be responsible for pushing the cell away from each other. And the engine's going to be in the middle, forming longer and longer spindles pushing out. So the kinetochores move and separate the sister chromatids. The polar microtubules are going to push the daughter cells away from each other. And then centered around our centrioles, we have these star-like fibers. See, that's where they get the astral, like the Houston Astros star. Well, the astral fibers, astral microtubules, are centered around our centrioles. And they're often illustrated just with, you see these little projections out there? Those are going to be our astral microtubules or astral fibers. So all of that was prophase. Our kinetochore and the spindles that are attached to our kinetochore and our sister chromatids, they push and pull until all 46 of our sister chromatids are lined up at the equator. And when they are lined up at the equator, we are in metaphase. Here's our illustration at the bottom. There's our actual photograph at the top, light level photograph. There you see our sister chromatids in the middle. You can see our spindle apparatus. There's our centrosome on either end, and you can see the fibers radiating out and away. Those are our astral fibers. Now, most of what they're illustrating are the spindles coming to the kinetochore, but you see other fibers sort of passing by. Those are going to be our polar fibers. So not all of the fibers actually attach at the kinetochore. So far we've got the P and the M of PMAT, prophase, metaphase. Now, as soon as we separate our sister chromatids into 46 individual chromosomes heading toward each pole, our kinetochore microtubules are pulling the chromosomes toward their pole. These are our polar spindle fibers that are pushing the centrosomes further and further apart. Now we are in anaphase. So basically, we're centering around metaphase. If you see them lined up, that's metaphase. If they are sister chromatids and they're not lined up, it's prophase. If you've got individual chromosomes not at the midline, we're in anaphase. See how easy that is? And then telophase is simply the last stage of the process. This is giving you a little example of the difference between our kinetochore microtubules and then our polar microtubules as we're doing the two things. We're pulling the chromosomes toward the pole and we're pushing the poles further away from each other so we have more space in the middle with which we can undergo cytokinesis or division of the cell. And for me, anaphase is fine. We don't have to understand A and B, even though both things are happening. Now, in telophase, this being the last stage of mitosis, this is going to be overlapping with the beginning of cytoplasmic division or cytokinesis. So once we see our chromosomes at each pole, once we see our nucleus beginning to form and our chromosomes beginning to relax, we're going to see the cell beginning to pinch itself off. That's the beginning of cytokinesis. So when you see the beginning of cytokinesis, you know you're in telophase. And in animals, we're going to form something called a cleavage furrow. It's like a belt around your waist that gets tighter and tighter and tighter until you pinch in half. And in plants, we're going to form a cell plate and a new cell wall that divides the plant cells into two different cells. And after telophase, what's the next phase that we're in? After telophase, we are in interphase. And what are the stages of interphase? G1, S, G2, and then we start prophase, metaphase, anaphase, telophase, and then interphase all over again. Yes, ma'am. Yeah. We've got the nucleoli. Okay, that's begin to form. They don't show it here, but here we've got the nuclear envelope in our illustration starting to reform. It's not going to really be completed until we completely separate and we're back in that G1 of interphase. All right, so if we put it all together, in animal cells, here we're seeing a nucleus, our DNA fairly dispersed, nice cytoplasm in the cell, that's our interphase. Now we're starting to condense. Do you see how you see the threads, thicker threads forming inside the nucleus? That's our sister chromatids condensing. They're sister chromatids because we've already come through S phase of interphase. We've made our copy. So we have our sister chromatids. We're not at the midline, so are we at metaphase yet? We have sister chromatids, so we must still be in prophase. Maybe it's later stage of prophase. Right along the midline. See, I like that. If I'm at the midline, I know that's the M phase, metaphase. Separating our sister chromatids into individual chromosomes. Anaphase. We've arrived at the poles. We're starting to relax. We're starting to divide the cell. Now we're at telophase. PMAT, the 4 stages of mitosis. We form 2 daughter cells. How do they compare to the parent cell? Identical. Absolutely identical. That parent cloned itself. In humans, how many chromosomes do we have in all of our body cells? How many chromosomes? 46. That's why I did that, and I'm glad you said that. We have 23 pair. So we have 46 chromosomes. You got a set from mom and you got a set from dad. And that's all you got from dad, right? Mom, you got everything else. You got all your mitochondria, all your Golgi, all your ribosomes. Yeah, dad just gave you a set of chromosomes. We have 22 pair that we call our somatic chromosomes. And our 23rd pair, that last pair, are sex chromosomes, either XX or XY. So that's your 23 pair, but 46 chromosomes. In mitosis, at metaphase, what is lined up across the midline? How many sister chromatids? 46. 46. Because, if you sit down and reason logically, you got 46 sister chromatids, they're going to get split in half. 46 chromosomes this way, 46 chromosomes the other way. When we get to chapter 20 in meiosis, we're doing something different. That's why we're hammering this sister chromatid part. And that numbering, 46. 46 sister chromatids, because this cell's got to have 46 chromosomes, and the other cell has to have 46 chromosomes. So they have to be the same in that number. Now, plants, it's going to look the same except for the cytokinesis part. So we have prophase. We still have prophase. See, we're real condensed here, but nothing's at the midline, right? If we're not at the midline, we can't be what? We can't be metaphase. Now, they're saying that's fairly along the midline. That looks kind of, I don't know. Maybe that's the best picture they could get. We'll call that midline. And by the way, you're not going to have to look and identify pictures. You're not going to look and identify pictures. They'll just be descriptions like all of our other tests. That's a little bit easier to see that we've moved the individual chromosomes apart. We can still see our spindle fibers in the middle. That's fairly easy to see anaphase. And here we can see our cell plate forming. In the midline, we've got our nuclei beginning to reform, our chromosomes beginning to relax, so we know we're in telophase. So here are our 2 types of spindle fibers. We're not going to talk so much about the astrals that have this star-like appearance, but there's our kinetochore spindle fiber attached to the centromere, and then we have our polar fibers. And do you see how we've got this this linkage that's attached here? As you add more and more microtubules to this point and you extend this chain longer and longer in this direction, do you see how that's going to push the centrioles and the poles further apart? If you keep adding more and more, and as you add, you push and add and push, it's like a conveyor belt, pushing in that direction. That's going to cause that centriole to be pushed apart. Now, likewise, whenever we have our kinetochore, we're not going to add microtubules at the end. What are we going to do here at the centriole? We're going to take away. And as we take away these monomers, these tubulin monomers of microtubules, as we take away, it's going to pull this shorter and shorter, like reeling in a fishing line. Think about these like extending one of those telescoping poles. So we're going to extend this pole from the middle and push out further and further. Here, we're reeling in by taking off so that we can separate these two and pull them in opposite directions. See how that works? So that's the engine that's going to drive moving the chromosomes and moving the spindle poles with our polar microtubules and with our kinetochore microtubules. And again, this is a pretty similar look to what we saw. This will be at anaphase here at the top. You can see that we've attached to those kinetochore proteins in the centromere, and we've pulled that apart as we're disassembling at that end. It's a fairly rapid process. When you start metaphase, there's a little bit of a pause, but then once they start moving, they're reeled in pretty quickly as the cell is elongating. So cytokinesis, this last step in the M phase. Remember, it's overlapping with telophase. So this is an animal cell. Do you see how we've got this called a cleavage furrow? It's this indentation around the middle of a cell. And in this case, this middle part of the cell primarily is comprised of a cytoskeletal microfilament called actin. Actin is abundant beneath the plasma membrane of our cell. But we've also got myosin that's involved. Myosin and actin together is what's responsible for you getting a muscle contraction, because these filaments can slide across each other. And what does a muscle cell do? It does one thing and one thing only. Contracts. It shortens. So as you have overlapping actin and myosin that forms a belt around this cell, as myosin and actin slide across each other, that belt just gets tighter and tighter and tighter and tighter until those two cells just pinch themselves off. It's almost like binary fission in bacteria. They just pinch each other off, and so you end up with two smaller cells half the size of that cell that it came from. That's why it's important for that G1 phase, for that cell to get big enough so it can divide. So that's the cleavage furrow made of our contractile belt of actin and myosin. Now, this is pretty interesting. This is one of the few times where you're going to get cell division without growth of a cell. And this is a fertilized egg. And that's why eggs are often some of the largest individual cells that you can find in the body. Because when you start off with a fertilized egg, you're going to begin to get divisions, many, many different divisions, until you have a hollow ball, which is called a gastrulous stage of embryonic development. But because these cells divide without increasing in size, these are called cleavage divisions. Now that's sort of a little freebie and a little trivia, simply because I love embryology and the study of how organisms develop. But that's one of the few times you're going to see the cleavage division without a cell going through this G1 phase without the growth. You're going to have to go through S. You've got to replicate the DNA. You've got to have the same number of chromosomes in all these cells. They just don't take the time to grow. You start growing after you go through a bunch of different divisions. Now, in a plant cell, this is one of those few places where I enjoy talking about plants, because it's got a cool word. There's some words in biology you just like to say, because they're just fun how you say them. When you look at follicular development in the ovary, fluid is going to build up inside of an ovarian follicle. And it's going to make that follicle get bigger and bigger and bigger. It's like blowing up a water balloon. Until you have one follicle that is so filled of this fluid, that one follicle is as big as the entire ovary itself. And this fluid continues to build up and build up until the fluid hydrodynamic pressure ruptures through the follicle and it ruptures through the wall of the ovary. That's what causes ovulation. And the fluid that's inside this follicle that builds up and builds up is called liquor folliculi. That's just fun. Say that. Liquor folliculi. It's like your tongue is just about liquor folliculi. That's just fun to say. Well, my word for botany is phragmoplast. Phragmoplast. That's just kind of fun to say. But what's going to happen in the middle of our daughter cells, where we're going to form our new cell wall, you're going to start forming these barrel-shaped structures called a phragmoplast. It looks a whole lot like your spindle fibers. And at the end of the phragmoplast, we are going to move material that are going to be formed, our components of our cell wall are going to be directed to the very middle. And it's in the very middle where we're going to form the phragmosome and the beginnings of our cell wall until we have laid enough material down that we've completely separated our cells into two individual daughter cells. So cleavage furrow in animal cells, that's cytokinesis. In our plant cells, that's going to be mediated by the phragmosome, which is composed of our microtubules, not called a spindle, but called a phragmoplast. Now, in plant cells, what moved the nuclei apart, what separated the chromosomes, that's still microtubules and spindle. But the division of the cell were these microtubules organized in a different manner to move our material for our cell wall, which creates the division of our two cells. So there we see our cell division. We've got our, see, and again, they've got all kind of crazy terms. Here's our pre-prophase, no, it's either interphase or prophase. Then here we have our chromosomes lined up along the midline, metaphase. We've skipped anaphase at this point. And there we have telophase, our material at each pole, chromosomes relaxing, and then we have our phragmoplast and our cell wall material being deposited. At the end, we get growth, and finally, we've got our new daughter cell that looks and is the same shape as the one we started with. And that's going to be happening in late interphase, late G1. When we look at cancer, if a lot of people find out you're biology majors, they're going to ask you a lot of clinical questions and a lot of biological questions and expect you to be an expert. So if someone asks you about cancer, what is cancer?
Mutation that keeps the cell growing continuously.
It's a mutation that leads to the loss of a cell dividing. Most of our cells in our body don't divide that fast. I mean, some of our fastest dividing cells are the cells for our hair, cells that line our gastrointestinal tract. They reproduce every 5 to 7 days. But most cells in our body don't divide that rapidly. So as a cell undergoes a mutation and loses the control of cell division and just divides out of control, that's the beginning of the form of a tumor. And if those cells leave and move somewhere else in our body, that then is the definition of cancer. But you can't get cancer until you lose that control of cell division. So our controls for cell division are fairly rigorous, fairly tight. We've got controls. Let's see, here's a restriction point between G1 and S. So when our cell is getting to this point before it starts synthesizing DNA, at our G1 to S checkpoint, they're like, okay, do we have enough organelles? Are we large enough in size? Has our DNA been damaged at all? It's kind of like the checklist at NASA before they launch, a new rocket. And if everything checks out, then the cell is allowed to move into the S phase. It undergoes DNA replication, forming our sister chromatids. We move into G2, and G2 we're preparing for mitosis. But before we move into G3 from G2 to M, okay, are we big enough? Have we replicated our DNA appropriately, and is there any DNA damage? You see the common theme? Is the DNA damaged? Is the DNA damaged? If the DNA is damaged, you're not going to divide. You're not going to make another messed up cell. And then our last checkpoint is metaphase to anaphase. This is the only one that happens during mitosis. And basically, this metaphase to anaphase checkpoint, and the reason we have the pause at metaphase, is have our chromatids attached to the spindle, to the kinetochores, and are they at the midline? Because if we have not attached 46 spindles on one side, 46 spindles on the other side, when we go to separate those sister chromatids, if one doesn't separate, then we've got a problem, because we're going to have two copies of a certain chromosome moving to one cell and no copies moving to another. That's called non-disjunction. So this M to A transition is checking for non-disjunction of our sister chromatids. Now, if that happens in a sperm or if that happens in an egg, and in most cases it's more likely to occur in an egg. If that happens to chromosome 21 and that egg gets fertilized, what is that baby going to have? Down syndrome. That's how you get an extra copy of a chromosome, because we didn't separate and we lost our control of the checkpoint. And unfortunately, in adults, I mean, it happens in men, but it's more commonly happening in women. The loss of that checkpoint happens in what case for the female? When are you at higher risk of having a child with Down syndrome? As you get older. So with age, it appears that these checkpoints are lost, and you can have more mistakes that happen because of non-disjunction and chromosomal anomalies as you get older. But again, that's with maternal age, the risk increases, but they're finding more and more that paternal age can also lead to chromosomal anomalies as well. Now, it's pretty cool, because another part of this control, moving from checkpoint to checkpoint, from phase to phase, it's regulated by various factors that sometimes we call CDKs and others we call cyclins. And there's one factor, maturation promoting factor. This was discovered in frog eggs. And if you were to inject this maturation promoting factor, so here we have a cell that's undergoing mitosis. If you were to take some of that cytoplasmic material and inject it into a cell that's not undergoing mitosis currently, that's going to cause the cell to undergo mitosis before it should. And what they found out was this maturation promoting factor is going to stimulate our CDK and cyclin complex that are on a timetable. How do we know when the timing is right? How do we know all these things? Well, it's these factors, the CDKs and the cyclins, that are very highly regulated, that then lead to the progression from one stage to the next. Now, have any of you heard karyotype? Have you heard that term before? Karyon refers to the nucleus. And in many cases, when you look at our chromosomes, they're going to be pictured like this, right? There are 22 pairs. Then we've got our 23rd pair, that's our sex chromosomes. But what are you actually looking at here? The copy that are still attached together. So are they chromosomes? Or what are they called? Sister chromatids. The reason that you're able to see these is they induce the cell to undergo mitosis. They arrest the cells at metaphase because they add a chemical called colchicine that dissolves microtubules. So the sister chromatids can condense, but they can't be pulled apart. Then you take that cell and you take a picture of the nucleus and you identify which chromosomes, based on their size in this case, or their shape, pair with which chromosome. You can also use fluorescent markers our fluorochromes. Remember how we talked about fluorochromes with light microscopy? You can label various little probes made out of DNA. And you can label them so that they bind to different areas, and so you match up the color coding of the chromosomes, sister chromatids, and you can align them so you can see if everything looks normal. Because in many instances, you might see, okay, we've got 2 chromosome 12s, but one of them might be missing a big chunk. So it's not enough that you have too many or too few entire chromosomes, but you can have some of these segments of chromosomes change places with other chromosomes. That could cause problems as well with the regulation. So that's one reason you look at karyotypes, just to see, okay, do we have all the chromosomes? Are they all lined up in the right place? Are they the same size? Or have we lost or gained any parts? So here we have our cyclins, our CDKs and our cyclins. When you look at our progression from mitosis through interphase and the stages, you can see that our maturation promoting factor, that's going to peak at the initiation of mitosis. But as mitosis progresses on, and before we even get to 1, that decreases. But then when you look at this mitotic cyclin protein, it's going to correspond as well with the peak of our maturation promoting factor. In fact, cyclin is on this delayed increase. And as it gets to a threshold, that's going to turn on the maturation promoting factor. And once it peaks, then cyclin goes, okay, I've done my job, let's do it the next time. So it's almost like a metronome. Mitotic cyclin and the level of expression of mitotic cyclin is very tightly regulated in a timing fashion. And mitotic cyclin's role is to activate maturation promoting factor that leads to the actual M phase. So here we've got a bunch of second messengers as we progress through. Here we have our cyclin CDK complex. All right, it's inactive. We've got to have enough of our cyclin around, enough of our mitotic CDK. But what else do we have to have? We've got to have ATP. Now, with our ATP, we've got to go through 2 rounds. This is an inhibiting kinase. What does a kinase do? Adds a phosphate group, right? So we're going to add this phosphate, and actually we were doing 2. But once we get over here, we've got this final kinase that adds a 3rd phosphate. We add that 3rd phosphate, we're still inactive, but that 3rd phosphate, that's the on switch. These 2 phosphates are going to keep it off. And what happens is we have got a phosphatase, which is the opposite of kinase. ACE is an enzyme. The first part's usually what's removed. In this case, we're removing a phosphate. So once we get an active complex, which means we have a positive feedback. And this active complex positively feeds back to this phosphatase to remove these 2 inhibitory phosphates that allows our active single phosphate in the right place to keep this guy going, that then activates our maturation promoting factor, and we get mitosis. So do you see the second messenger signaling here? We get the cooperation of these. We have 2 kinases. We have a phosphatase. When we link all this together, now we can progress through to actual mitosis. So our cell cycle regulation, all these checkpoints and stops and progressions through. As we go through our mitotic activity, we see our G1, we see our G, we see our G2. We can see that we have our mitotic CDK. We're going to begin to form that. It's dissociated from everything else. We're going to have that in G1. It progresses around through S. But we don't join our cyclin to our CDK until we're into G2. and even late in G2. And once we get into our late G2, we get to that transition. That's when we add the phosphates. Then finally, we remove those 2 inhibitory phosphates to turn it on and progress through mitosis. And that's the end of mitosis. That's when it's probably in anaphase. We begin to break down the cyclin. It gets degraded, probably by another enzyme. And when you degrade the cyclin, that turns it off, and we've still got our CDK that comes back around, picks up another cyclin, and it starts the process again. That's why in our graph that showed cyclin up and then drop way down, it dropped way down because we're degrading it, and it just takes time for the cell to make more. And it's that timing and the synthesis of the cyclin then that is primarily controlling the timing of our cell division. It's like the CDK is just a component that's recycled all the time. And the cyclin is going to be the key to get it started once we add the phosphates. Ooh, is that complicated or what? That's putting it all together. This is just a summary. You don't have to know all these steps. What we just went through, that's what I want you to know. The relationship between CDK and cyclin. So there's our cyclin, there's our CDK. This is just showing those kinase activities that are happening when you add the phosphates to everybody, and then you come back around.
What was the thing that broke down the cyclin?
It's going to be an enzyme. They don't list the enzyme.
I think it was listed, something was listed on the previous slide, but anaphase promoting complex? Is that,
Yeah, not just, I guess it's
so it's not?
it's an association of proteins, but there's got to be one enzymatic factor, and we don't know the name of it. We just know it is broken down. I mean, there's probably some primary research articles that's got that listed, but for our purposes, no, we don't have to put a name to that one.